Welcome back, science enthusiasts. Today, we're diving into the fascinating world of cellular respiration, specifically focusing on a crucial aspect known as the Krebs cycle. So grab your lab coats and let's explore the intricate steps of this metabolic pathway. Before we delve into the nitty-gritty details, let's understand the significance of the Krebs cycle. Also known as the citric acid cycle or the tricarboxylic acid (TCA) cycle. It's a central part of aerobic respiration, occurring within the mitochondria of eukaryotic cells. Its primary function? To generate high-energy molecules such as ATP and NADH that fuel various cellular processes. Our journey begins with the entry of acetyl-CoA into the cycle. Acetyl-CoA is derived from the breakdown of carbohydrates, fats, and proteins. It enters the cycle by combining with oxaloacetate, forming citrate in the process. Next, citrate undergoes a series of transformations, ultimately leading to its conversion into isocotrate. This step is facilitated by the enzyme aconitase. Isocotrate is then oxidized, resulting in the release of CO2 and the formation of acetoglutarate. Simultaneously, NAD+ is reduced to NADH. This reaction is catalyzed by isocotrate dehydrogenase. The next step involves the oxidative decarboxylation of acetoglutarate, producing succinyl-CoA. Once again, CO2 is released, and NAD+ is reduced to NADH. This process is mediated by the enzyme acetoglutarate dehydrogenase complex. Succinyl-CoA then undergoes a substrate-level phosphorylation reaction, leading to the formation of succinate and the production of GTP. This step is catalyzed by succinyl-CoA synthetase. Succinate is oxidized to fumarate, resulting in the production of FADH2. This oxidation step is facilitated by the enzyme succinate dehydrogenase, which also serves as a part of the electron transport chain. Fumarate is then hydrated to form malate, catalyzed by fumarase. This step prepares malate for the final transformation in the cycle. Finally, malate undergoes oxidation to regenerate oxaloacetate, accompanied by the reduction of NAD+ to NADH. This reaction is catalyzed by malate dehydrogenase, completing the cycle. And there you have it, folks, the intricate dance of molecules within the Krebs cycle. Through a series of 8 enzymatic reactions, cells efficiently harvest energy from carbon fuels to sustain life processes. This metabolic pathway not only generates ATP, but also provides intermediates for biosynthesis. Truly, the Krebs cycle exemplifies the elegance and complexity of cellular metabolism. Thank you for joining me on this scientific adventure. If you enjoyed this video, don't forget to like, share, and subscribe for more exciting content. Until next time, stay curious and keep exploring the wonders of science. Take care and see you soon.